Good morning, everyone. My name is Rona Varghese,
And I'm Hannah Pulliam.
and we're from the University of the Sciences in Philadelphia. And today we're presenting our poster, Shining a Beam of Light on Phages. The novel bacteriophage beam replicates in host bacteria Mycobacterium sphygmatis, and it's found in a direct soil sample with the morphotype Sypho verde.
So we sought to annotate Beam's genome and assign functions, as well as create a phylogenetic tree with other cluster J phages and compare this to the pH of their soils.
Beam was found to have two terminate small subunits, and it was also a lysogenic phage found in cluster J with an unusually large genome. So for our materials and methods, the manual annotations and function assignment, we use DNA master, GeneMark, BLAST, FAMRate, and HHPred. For our bioinformatics portion, to make phylogenetic trees, we use Cluster Omega Microsoft Word. And for soil acidity, we used USDA Web Soil Survey, which had information of soil types, specifically the pHs of them. So for our first portion of the results, phages like Z-Link were found to have the most common genes, similar to phage B. And Z-Link is found in cluster J, and so with other cluster J phages that had common genes with Beam, we made a cladogram phylogenetic tree showing those evolutionary relationships to a common ancestor. And the numbers represent the length of the branches, and the larger the number, the greater the genetic change of the phage from the common ancestor.
So for our soil results, what we did is we used the coordinates from phages CV for each phage and plugged them into the USDA Web Soil Survey algorithm. As you can see in that top figure, that is what our information looked like with a local soil type. So Beam was found in a soil of 5.3 pH, which is classified as highly acidic by the USDA. We also, with our randomly selected cluster J phages, we found that they also existed in highly acidic or slightly acidic soil, with aerial being the outlier. So that bottom figure shows a graph of the pHs in order of evolutionary distance from the common ancestor. So you can see there's a negative parabolic relationship in that. So for our annotation results, we found that there were two small terminate subunits that were encoded for in gene products 1 and 9. We also found gene Pulliam was a DNA methylase. So as you can see in the figure above, in the auto annotation box, there are 4 other possible genes that DNA master called for. However, they did not provide any blast hits or were in an ORFAM. So you can see in the right figure that gene Pulliam was manually annotated for and saw strong hits for DNA methylase in both HHPred and BLAST.
So for our conclusion, like what Hannah said before, there was 2 terminate small subunits in Beam, and this could be because of Beam's large genome. There had to be a second terminate small subunit in order to package the entire viral genome. And cluster J phages also had an average pH of 5.2, which is strongly acidic. And finally, gene Pulliam was overtop in ORFAM, and gene 22 was in that ORFAM and called for DNA methyltransferase.
So some future experiments, we would decide that we would have spliced out gene product 1, which is one of the small terminate subunits, and amplified it in a host cell to see how its replication is affected, to see if that second terminate small subunit really played a part in replication. We would also conduct experiments with cluster J phages in different environmental concentrations of pH to find its optimal pH for the Sypho verde phages. We also decided that we would see how Beam's long genome may help or hinder medicinal purposes. So how we would do that is we would run the same medicinal experiment with cluster J and cluster A phages, which are known to be much shorter, and see if there was any difference in its effect.
We would like to thank our youth sciences faculty and our C Phages teaching assistant program, and a special thank you to our professor, Dr. Sonnen, and our presentation author, Malia Hart. We couldn't have done it without you.
We would like to thank you for coming today and hope you enjoy the rest of the symposium.